innate immunity. And I didn't quite get to finish what I wanted to cover, which is about the inflammatory response. This is the part in the book at the end of the innate immunity section of the book that's addressing it. But I want to take a second to sort of step a little bit outside of what the book is saying to address kind of a more classical representation of what the inflammatory response is about. So this is a picture that I'm actually familiar with because when I was in graduate school, one of my roommates was a medical student, and in the physiology class that he had, this was the picture that was used to represent this. It's sort of a way of describing this, been around for a long time. Inflammation, the inflammatory response, is a series of reactions, not all of which always happen every time this occurs, but these five characteristics are associated with inflammation. And in the center, holding it up mostly, is swelling. The inflammatory response is really about the accumulation of fluid at a site of infection or tissue damage or something like that. It happens because blood vessels dilate and the tissue is perfused with blood more. The blood vessels, when they dilate, they also become a little leakier than normal, and so more stuff comes out. And beyond just the normal fluid that's going to be accumulating in tissue, like we've already discussed, setting up the reason for the lymphatic system, this is going to increase that fluid that's accumulating. The lymphatic system is then going to have to drain that tissue, that fluid out of the tissue even more, which lends itself to being part of the immune response. To some degree, from that accumulation of fluid, because of the increased perfusion of the tissue, the other four kind of fall in with that. The two on one side and the two on the other side are kind of separate in what they have to do with it. Now, on this side, next to swelling, is redness. It's represented with the character whose toga has fallen off or whatever, and so he's embarrassed and his face is turning red. The reason why his face is turning red is the exact same reason why there's redness associated with swelling. There's increased perfusion to the tissue. The embarrassed reaction is just something keyed into an emotional reaction, but it's the same reason. Just an accumulation of blood in the tissue is going to turn the tissue red because there's going to be more hemoglobin there. And then next to that is heat, which again can be directly related to the increased perfusion. As blood is accumulating in tissue, then that tissue is going to be a little bit warmer, just because your body temperature is really established by the temperature that we see in blood as it moves through the body. And anywhere in your body that blood accumulates, that's going to make that tissue warmer. There are also chemicals that can be released as part of the inflammatory response that can specifically raise temperature in tissue. The main way that temperature changes in tissue has to do with mitochondrial activity. Mitochondria break down molecules to make energy through a series of processes that are exothermic. They give off heat. So anytime that we need to see an increase in heat, there could just be an increase in metabolic activity in the mitochondria. There's some suggestion actually that mitochondria can preferentially be inefficient in their chemical reactions and give off more heat than they would normally do, which is just going to help raise the temperature of tissue. On the other side, also related to the accumulation of fluid, the swelling that we see, we're going to see pain and loss of function. Now, as tissue swells, there's going to be pressure put on certain structures, such as nerve endings, and so that can generate some pain. But there's also chemicals that are released as part of the inflammatory response that are specifically going to activate pain fibers. I talked about one of those when I was talking about hormones, and I told the story of aspirin and Vioxx and prostaglandins and that sort of thing. So prostaglandins that are made by cyclooxygenase 2 are specifically part of the inflammatory response and activate pain fibers. And so by taking a drug that blocks the cyclooxygenase enzyme, like aspirin, then you can decrease pain that's associated with that. Now, pain actually is mostly an indicator of tissue damage or infection, so that you know to treat that part of your body a little bit more carefully. It's not associated with pathogenetic infections, but the example I like to use is, imagine if you've sprained your ankle. The swelling around your ankle is going to be inflammation. The pain associated with that is going to make you not want to put weight on that ankle, and so you'll favor the good ankle, allowing the sprained ankle time to heal a little bit more. And so the pain is there for that. If you've cut your thumb or something like that, and where the cut is, it stings a little bit, you're going to be careful not to move the thumb too much and stress that tissue. You're not going to grasp things and potentially activate more pain fibers. So it helps you to leave that part of the body alone a little bit more so that it can heal up. The last thing there is loss of function, and the sprained ankle idea kind of goes with that too. Again, the swelling, the accumulation of fluid at the sprained ankle is going to limit the amount of movement that's possible at that joint. So there's loss of function for the joint. That's, I think, a fairly easy example of what that can mean, but you'd see that a lot of different places. So if you have pancreatitis, and the itis ending is referring to inflammation of the pancreas, then the pancreas isn't going to be able to do its job as well as it's supposed to, which is largely going to affect the secretion of the digestive juices coming from the pancreas. I'm not that familiar with pancreatitis, and I don't know necessarily if it's going to affect insulin and glucagon aspects also, but that's minor to what the pancreas does. So it's probably not going to be mostly affected. But that's the idea, that when an area is inflamed, there's going to be some loss of function because that tissue has to spend some time and energy in trying to repair itself. So it's not going to work quite as well. So that's sort of the overall idea of what we think of as the characteristics of the inflammatory response. If we go to the picture from the book, this is more about what's the point, or not what's the point, but what's happening in the inflammatory response. So here we have a wound of some sort, a cut through the skin, and pathogens are entering in through the skin because as a barrier, it's no longer effective. It has a big hole in it. With the damage, actually, when cells are broken open or damaged to some degree, they're going to start releasing chemicals which will initiate the inflammatory response. Also, there are mast cells in the area, which are characterized in this picture by these two kind of purpley looking things here. Mast cells release histamine, and histamine is specifically going to activate the blood vessels, and those will then dilate and become leakier so that tissue can perfuse into, fluid can perfuse into the tissue. Okay. Along with that, white blood cells in the bloodstream will be able to move out of circulation and into the tissue. Again, because they have, because of tissue, because the blood vessels have become leakier. Now, if you look at the white blood cells represented here, and granted they're not that easy to see in the picture. Let me enlarge the picture a little bit. So I want to just point out in the white blood cell here, its nucleus looks very unusual. We tend to think of nuclei as being very spherical in appearance, but with white blood cells, they tend to have this kind of sort of lobular structure to them. The other one represented in this picture, there are little sections of the nucleus that are linked together by little filaments. All of that is the nucleus, it's just not very spherical. What this allows for is this cell can, the picture doesn't actually depict any openings between the cells, but where two endothelial cells come together, there's a little cleft. And when histamine causes dilation of the vessel, those intercellular clefts are going to be a little looser and leakier. And so the white blood cell will squeeze through there. Its nucleus looks like this because it's easier to shove a little bit of the nucleus through at a time rather than trying to get a big spherical nucleus through the gap all at once. So when we see this sort of nucleus in a white blood cell, it actually is an indication that the white blood cell is optimized for that movement through the intracellular gaps in the endothelium. And so they'll move out into the tissue, which is represented in the second frame here. And when they're at that point, they're going to become phagocytic. So these are probably neutrophils represented here, although it's not clear from the picture exactly what they're supposed to be. But it doesn't really matter. It's one of those types of white blood cells that are phagocytic. And then when they move out into the surrounding tissue, they're going to be able to get rid of the bacteria that might have come in, these pathogens that they suggest in the picture, as well as any debris from the damage. So there are going to be cells that have been damaged in the wound, and so those cell fragments that might be left over from that can also be phagocytosed and cleared out of the area. Okay. So in this picture, they don't really highlight all of the kind of things I just mentioned, but there's definitely some accumulation of blood in the area, which is going to make the tissue red in color. And then there's no way to depict it in a picture, but there's probably going to be activation of pain fibers because prostaglandins are released in the area and that sort of thing. And you can imagine if you get a cut through your skin, it usually does hurt a little bit. We can see in the picture that there's sort of a bump around the wound, suggesting that it's swollen right at that particular area, that sort of thing. There's a little bit of a reddish glow here, which I think might be representing temperature increasing locally right there. And that change in temperature can help to inhibit bacterial growth. The bacteria that want to get into our body want to live at 37 degrees Celsius, which is body temperature. So anytime that body temperature goes up, it helps to restrict bacterial growth. When we have a rather severe infection, then we'll have a systemic change in temperature, which is what we call a fever. And the point of that is to really make your internal environment as unpleasant as possible for whatever pathogens got into it, trying to get rid of it. If it isn't very effective and the bacterium or whatever persists for a long time, then you'll have a fever for a long time, which can then create problems because your body is optimized for working at 37 degrees. And if it spends its time too high, 42 degrees Celsius for a long time, that's going to start affecting the tissue in your body, especially brain tissue, and there'll be some really severe consequences of it. But changes in temperature, whether it's local in the inflammatory response or systemic, as in fever, are meant really to inhibit bacterial growth to aid the immune system's ability to get rid of that infection. Pleasure. So that's the inflammatory response there. Just a little bit said in the book, but I wanted to use that other picture to kind of characterize a little bit more. Okay. Now, as far as the chapter is concerned, we looked at the structure of the lymphatic system before, and then last time, and I just had to wrap it up here at the end, we're looking at innate immunity, which is what this section here has been about. And now we want to move on to adaptive immunity. And this is potentially a rather troublesome concept. And so I want to present it a little bit differently than the book does. I'll keep mostly to the way that the book's laying it out, but I want to start it out by kind of pulling a couple of things together that are a little bit separate in the book. Now, the point of the adaptive immune response is that we're fighting off specific infections or specific threats to our internal environment. What we've talked about before, phagocytes, natural killer cells, complement, great. What was that viral thing? I'm blanking on the word now. What's the signal from a virally infected cell that I talked about last time? Starts with an I. Interferon, that's it. Those sorts of things are very general responses. Now, interferon is specific to viruses as opposed to bacteria or something like that, but it's not specific to one particular virus over others. The adaptive immune response is about recognizing specific pathogens, recognizing the influenza virus rather than E. coli or whatever it might be, being very specific about what you're responding to. And the way that it does that is because there are these specialized molecules that recognize very specific molecular structures. One of those is what's represented here, and this is a picture from a little further down in the chapter from where I just was on the page. This is called a Tcell receptor, okay? And so we see it here, it's embedded in a cell membrane, which we would expect for receptors. And I just want to point out here that it has two halves, which are basically identical. There's the alpha chain and the beta chain. And by identical, I mean structure-wise. And each one has a constant region and a variable region. Okay? Now, I want to move from this to another picture, and this is actually from later in the chapter where they're talking about B cells instead of T cells. But I want you to see how the two are related. So this is a T-cell receptor, and this other picture I want to pull up is an antibody. Now, if I could put these two pictures side by side, which I guess I can do. Would help to be there. Okay, so in the T-cell receptor, we have a variable region and a constant region in the part that is transmembrane. And in an antibody, we have a variable region and a constant region, which includes all of the rest of this. I'm not saying it's necessarily the way that people tend to think of it, but maybe the outer two parts, the variable region and the first part of the constant region in the antibody, are similar to the variable and constant region represented here, and then the transmembrane region is kind of similar to the rest of the constant region here. I'm not saying that's necessarily the way it is, but it's a fair way to think about it for the sake of what we want to talk about here. Now, the variable region in either of these are called variable because from one lymphocyte to the next, whether it's a T cell lymphocyte that's making the T cell receptor or a B cell lymphocyte that's making the antibody, each individual lymphocyte, when it's first generated in the red bone marrow, rearranges that part of the gene for this protein a little bit, and it does it randomly. So each cell does its own rearrangement to make a unique variable region. And in that variable region, it's really highlighted in the antibody picture here. There's a particular shape at the outside edge, what's called the antigen binding site. That's what actually binds to whatever's gotten into your body. The word antigen is just very general, anything that generates an antibody response. That could be a virus particle, that could be a bacterium, that could be a foreign molecule that's gotten into your body. It could be a molecule that's supposed to be in your body. We call those self-antigens, and we'll talk about those specifically later on. But there's an antigen binding site. Now, they don't represent it in the T cell receptor here, but the same sort of thing's going on. The T cell receptor has to be able to bind to the antigen also. They do represent that binding in this subsequent picture here. So this kind of curly ribbon looking thing in the middle is some protein, some antigen. And we have 3 different T cells that are each binding to that antigen in the center, binding to different spots on it because they recognize that particular area of the antigen. Okay? And each spot where the binding's taken place is labeled as an antigenic determinant, which just means that's the shape of the protein that reacts with the binding site on, in this case, the T cell receptor. The same thing would be true of an antibody. So where the antigen binding site is, it's going to bind to an antigenic determining site on whatever it's binding to, whether it's a protein or a virus or a microorganism or whatever. Okay. It's going to bind to that. So the same sort of thing is happening in antibodies as well as T cell receptors. They work on the same basic principle. They are molecules that have a variable region, and that variable region uniquely recognizes some antigenic determining site on an antigen. Now, we don't know when we're born what antigens we're ever going to face in our bodies. So the process basically makes sure that we can recognize effectively every antigen out there that we might run into. It's not true, because there are limits to this, but ideally you're making an infinite number of antibodies or T cell receptors that are going to recognize anything out there that you might interact with. Then, where are we? You want to test out this lymphocytes that are making these T cell receptors, and something similar applies to the B cells that are making antibodies. You want to test them out to be sure that they do their job properly so that you'll actually have an appropriate immune response, and that you can you won't react to anything that's in your body, any of your self-antigens. And so that's what this picture is getting at. Now, this is concentrating on T cells, but some of what's going on here actually applies to B cells also. Not quite all of it, but some of it. The thing about T cells is that they're characterized as being cell-mediated immunity, which means that they interact with other cells. And so for them to work properly, there has to be a cell that they interact with. And what we call that cell they interact with in a very general sense is an antigen-presenting cell. I guess that should be hyphenated. Now, antigen-presenting cells can be of various types. They can be phagocytic cells. They can be dendritic cells, which is actually the type that the book tends to use as an example. Or they can be body cells. And for body cells, either they're going to be infected, meaning that they present the infectious agent, or they can be normal, which means they present self-antigens. Now, that's a little oversimplified, talking about body cells infected versus normal, but normal cells not being infected are not going to be able to present antigens that are part of an infectious agent, whether it's a virus or a bacterium or whatever. And then normal infected cells might present self-antigens also, but what's important is that they're going to have some evidence of being infected. Okay. Now, let's see. Here's a picture representing an antigen-presenting cell. This is a dendritic cell. What this is representing could almost just as easily represent a phagocytic cell, but not a body cell. So over here we have some pathogen, say, some microbe of some sort. It gets engulfed by this cell, kind of like the phagocytes that we saw the videos of last time. That ends up being, oh wait, that's not where I want to point. Over here, here's where the pathogen is labeled. So it gets engulfed into the cell and becomes contained within a little vesicle. Okay. And then for a pathogen, that vesicle merges with a lysozyme, which is the digestive organelle inside of a cell. There are digestive enzymes there, as well as it has an acidic pH, which ends up then digesting the pathogen. The bits and pieces of the pathogen that are left over combine with a molecule called MHC. Don't worry about what MHC stands for. When it was originally named, it was named based on what we understood it to do at that time, but so much has been discovered about what MHC can do that its abbreviation is really how it's referred to. The same way that when you go get a bucket of chicken, you're getting it from KFC, okay? It's no longer actually called Kentucky Fried Chicken because the marketing department decided people don't want to buy fried food, so you just buy KFC. Okay. So officially, that's KFC. That's how we refer to that. Okay. Same way, this is MHC. That's how we refer to it. I don't even want to go to explaining what the abbreviation stands for. When MHC and the fragment of the antigen come together, they get inserted into the cell membrane so that that little piece of the antigen can be presented to a T cell. Okay, that's why we call it an antigen presenting cell. Okay. Now, like I said in the book, they're using dendritic cells as kind of their go-to example. What I just described can, to some degree, be exactly the same as what happens in a macrophage or some other sort of phagocytic cell. I mean, really what we see here is phagocytosis. We just don't tend to really associate that with dendritic cells. Dendritic cells are mostly about presenting antigens of things that have gotten in through the skin or mucous membranes. Okay. In body cells, proteins that are being made in that cell are always being presented on the surface with MHC. Okay. Now, one difference is with the phagocyte or dendritic cell, MHC that's being used is MHC2. And since there's an MHC2, that should suggest to you pretty obviously that there's something called MHC1. MHC1 is used by body cells, MHC2 is used by phagocytic cells. So in your body cell, MHC1 is doing the same sort of thing, but with stuff that's being made in that cell. If it's a normal cell, it's making what it's supposed to make, which might be collagen or neurotransmitter receptors or blood type antigens, whatever it might be. Those things are being made normally. They get presented on the surface of the cell, basically as a little sign to the immune system that that cell's normal. It's doing what it's supposed to do. But when a body cell's infected, say with a virus, it's going to be making copies of the virus. So bits and pieces of the virus are going to be presented on the surface of that cell. But since the virus is not something that's supposed to be in your body, then it basically marks that body cell as being infected, and the immune system can deal with them. So the antigen that's presenting on the surface is there so that the T cell can interact with it. The Tcell receptor binds to the little bit of antigen that's attached to MHC there. Okay. At this point in the picture, that's about all that they're pointing out. This little thing next to the Tcell receptor, this little purple stick, plays a role too, which I'll point out in a little while. Okay. So, again, we're talking about T cells, which their official name is thymocyte, referring to the thymus, and that's what the T in T cell refers to. They're made originally in the bone marrow, just like every other blood cell. They move from the bone marrow to the thymus, and the T cells are the only ones that do that. B cells stay in the bone marrow, and that's why we like to associate bone marrow with B cells, although that's not actually what the B stands for. Now, the T cell has to go through a couple of tests, which are called positive selection and negative selection. Now, a little bit about the notation of what's going on here. Next to the initial thymocyte, in parentheses, they have two dashes, which means it's sort of a baby thymocyte, nothing going on yet. But then what's going to happen is it's going to eventually be able to produce and express a protein called CD4 or CD8. So up here, there's nothing, which is illustrated a little bit better down here. The two dashes are really minus signs. These cells do not have CD4 and do not have CD8. So they're CD4 negative, CD8 negative. Okay. Later on, we're going to see a positive sign saying that it's a CD4 positive or a CD8 positive cell. Okay. So before they express the CD4, CD8 proteins, they go through positive selection, which is simply that the T cell, thymocyte, interacts with MHC with antigen-presenting type cell. Okay, if it can make that connection fine, then it'll move on. If it can't, if the early T cell cannot bind to MHC, that means it's never going to be able to do its job properly. So it's going to be destroyed. Okay. Positive selection means that we select for things that are able to pass the test, and they go on. And the things that fail the test are destroyed. Then the next step after that early T cell recognizes MHC, it goes on to the negative selection stage. And with negative selection, at this point, the cells are going to be CD4 or CD8 positive. Okay? And we can refer to them as CD4 cells or CD8 cells. Now, in this picture, they're kind of throwing a few things together. They're oversimplifying the process a little bit, but we're really going to end up having cells that are just one or the other. And in this picture, it looks like there's a couple of things on the surface, but really there should be just one thing that we see in a couple of other places here. Now with the negative selection, the cell, we already know that it can interact with MHC, so it's going to be able to do its job properly. So now it's being tested to see if it recognizes self-antigens. Okay, so now it's presented with antigens by antigen-presenting cells or body cells. Specifically, it recognizes MHC, and we know it does because it passed the first test. If it recognizes the self-antigen, if the T-cell receptor binds to the antigen, then it's destroyed. Okay? Because if the T-cell receptor recognizes the antigen at this point, it's recognizing a self-antigen. This is not meeting something that's been presented representing infection. This is early on in development. So the cells that react to things at this stage are reacting to things that are supposed to be in your body. So we destroy those. And anything that does not interact with an antigen will survive this round of testing. Okay? And that's why it's called negative selection. We select for things that have a negative response. We don't want them to recognize self-antigens, because then that means what survives this only recognizes things from outside of our body. Okay? Now, we don't know what might be from outside our body that we're ever going to interact with, but this is a way to be sure that we're only going to have cells that can do that. After going through this, these cells will end up maturing into two types of T cells. There's the CD4 cell and the CD8 cell. The CD4 cell is going to eventually mature into what's called a T sub H cell, or the H there stands for helper. That's going to be a helper T cell. And the CD8 cell becomes the T sub C cell, which is cytotoxic. Now, helper T cells help other cells get activated. Cytotoxic T cells kill cells. Cytotoxic means cell killer. So they have different roles to play, but they're both T cells. Now, the mature helper T cells and mature cytotoxic T cells move out of the thymus and into somewhere else in the lymphatic system. So for the picture here, they have a lymph node, but it could be anywhere in the lymphatic system other than the thymus or the bone marrow. Now, how are thymus and bone marrow different from all of the other lymphatic organs and tissues? What's different between bone marrow and thymus and all of the other lymphatic organs and tissues? Well, bone marrow produces blood cells, yes, but that's what it does as part of the cardiovascular system. What I'm asking is, what's bone marrow and the thymus? How are they different from the rest of the lymphatic system? When we were talking about the lymphatic system, I pointed those two out specially and assigned them a name, then everything else had a separate name.
Primary organs?
Right, they were the primary lymphoid organs or tissues. Bone marrow isn't really an organ, it's a tissue. But remember this, primary. Bone marrow, thymus, okay? And then everything else falls under the group that's secondary, okay? The reason those are called primary is because they are where these processes take place. Okay, the bone marrow is where everything's born. The T cells go onto the thymus, the B cells stay in the bone marrow. B cells do not go through anything like positive selection because they don't interact with MHC. Only T cells do that. But then T cells and B cells are tested to see if they react with self-antigens, negative selection. T cells do that in the thymus, B cells do that in the bone marrow. Okay. And then they move on into the secondary lymphoid organs and tissues. Then they're ready to be activated. Okay, this is a mature cell, but it's not going to do anything until you are infected by something. Okay. But this is how we get to the point of having these mature cells here. And I just realized that the screen doesn't go down all the way. So I've been pointing down here at the mature cells, and the picture at the bottom there is a secondary lymphoid organ. It's a lymph node for the sake of the drawing, but it could be anywhere. It could be the spleen, could be a tonsil, could be mucosa-associated lymphatic tissue, whatever it is. They hang out there. Okay. Once they are mature and they're hanging out in the lymphoid tissue, then they're going to go through an activation process. Okay. When you get sick, that initial exposure is going to activate these cells, and they're going to be ready to fight off the infection. It just takes them a little bit extra time. Okay. So, what I've talked about so far, I'm just going through the chapter to some degree, and it's talking about antigen-presenting cells and differentiation of T cells in the thymus. I have this picture up already. There we go. Then we have one of these cells. It's a mature cell, but here it's referred to as a naive cytotoxic T cell, which means that it's never interacted with whatever antigen it's specialized for. Okay. So at some point, it's going to interact with the antigen that it's specific for. With cytotoxic T cells, they do that with antigen-presenting cells that use MHC1. Okay. Earlier, the picture I was showing you with the antigen-presenting cell just showed MHC2. MHC1 is specifically for cytotoxic T cells. And the reason that's specific is because of that CD8 protein. CD8 recognizes MHC1. Okay. What do you think CD4 recognizes? CD8 recognizes MHC1.
MHC2.
Yeah. CD4 recognizes MHC2, which will be relevant in a little while. Okay. When the naive T cell recognizes an antigen through its Tcell receptor, and CD8 recognizes MHC1 from the antigen presenting cell, then it's going to end up getting activated. Activation leads to this cell dividing, proliferating, and it differentiates into two different types of cells. One type is the activated cell, and it's the cell that's going to go out and do its job. The other is a memory cell. The memory cell doesn't do anything right now. It's just there, so when you're exposed to the same thing in the future, you already have cells ready to fight off the infection. Okay? The memory cells, the next time you're exposed to the same pathogen, will then differentiate into activated cells and memory cells, and you go through the same sort of ending part here. But you don't have to go through the initial exposure and activation. The cells are already activated, they're ready to do their job in the future. Activated cytotoxic T cells then hunt down cells that have been infected by whatever has gotten into your system. If it's the influenza virus, they go hunt down the mucosal lining cells in the respiratory tract, if it's a respiratory flu, or the digestive tract, if it's a digestive flu, and destroys the infected cells so that the virus can't replicate any further. Okay. If they're activated because of the presence of cancer, then they're going to hunt down the cancerous cells and fight them off, that sort of thing. And the memory cells are going to stick around for that later on, if it comes up again. Oh, whoops. This is activation of a helper T cell, okay, which I probably could have showed first, but the helper T cell is going to interact with an antigen presented by an antigen presenting cell with MHC2. And so CD4 and MHC2 are going to interact together. Activated T cells also differentiate into the active ones, and there are memory helper T cells for future infections. Activated T cells start to release chemicals called cytokines, which are basically hormones that are shared between white blood cells. And the cytokines will then help increase the activity or the activation of cytotoxic T cells, as well as B cells, which we haven't seen yet, but helper T cells help activate the other cells. Okay. And then this is just demonstrating sort of the same thing that was suggested in this picture here, that cytotoxic T cells then go out and destroy infected body cells. So this little section of the book here, right after the cytotoxic T cell picture I just showed you, talks a little bit about the proliferation and differentiation, which in the book they use selection and expansion, but it's the same idea. And then memory cells are there to help with future infections. The helper T cells release these cytokines, which are then going to affect cytotoxic T cells, another type of T cell referred to as a regulatory T cell, as well as B cells, which are mentioned down here. Now, I want to point out regulatory T cells for a second. Okay, so helper T cells are T subscript H. Cytotoxic T cells are T subscript C. And those are the main two cells that we usually think of. But regulatory T cells, which is T with a subscript REG, are an important type of cell, and we've met them before. These are the lullaby cells that came up in the Jasper Lawrence story that we listened to. Okay. They used to be called, and I don't quite know why the name changed, they used to be called suppressor T cells, because that's what they kind of do. They suppress the activity of the other T cells and B cells to some degree. So attack cell, as we met that in the Jasper Lawrence story, really kind of refers to any of these other cells, primarily the cytotoxic T cells, because those are the ones that really go out and attack things. But the lullaby cells are these regulatory cells which keep things under control. Now, I want to move away from the book for a second because there was something that that was shared with me recently, which I think is pretty good. I put this in the video content. I did show you the white blood cell thing last time, right? Yes? No? The little video of white blood cell chasing down a bacterium?
Yeah.
Okay, good. Which is this one right here. There's also a video that I've added in here. Oh, shoot. Sorry, I didn't put it in yours. I saved it in a different class. So let me just go there real quick. Okay, so there's what I showed you before. This new thing that I came across, where'd it go? There we go. As for a YouTube video that I could embed here, I put this one here, which is by the same people, but I'd rather show you the video that's in this web page right here. So let me go through this. I don't necessarily know what LabRoots is, but there's this nice video here that I want to play real quick. And this is of a cytotoxic T cell that's going to be destroying, I think it's a cancerous cell in the video here, but it could be just as simply an infected cell, you know, that's picked up a virus or something like that. Now there's some voiceover here, so I'm just gonna let it play out so you can see this, but the video showing the cell moving around is really quite incredible. And there's some credits at the beginning. Oh, shoot. Hold on a second. I'm used to another room I use where the speaker's in the projector, so it always plays. But.
These are immune cells called cytotoxic T cells. Cytotoxic T cells are able to recognize and kill virally infected and cancerous cells in the body. These T cells are on patrol, searching for their targets. Let's take a closer look at how they move. Cytotoxic T cells are constantly migrating around your body. They move through your tissues by pushing out the membrane at the leading edge of the cell and pulling themselves forward. When a T cell encounters a cancer cell, an explosion of membrane protrusions explore the surface of the target. Cytotoxic T cells kill their targets with toxic chemicals. The chemicals are housed in these red structures called lytic granules. You can see the lytic granules polarize from the rear of the T cell to the blue cancer cell. These toxic granules secrete specifically toward the target, protecting innocent bystanders which may be nearby. As the membrane of the target is compromised by the toxins in the granules, our indicator flashes bright red, signaling the impending death of the cancerous cell. Movies like these allow us to study the efficiency with which cytotoxic T cells operate. Now we'll leave you with a few more movies of T cells doing what they do best, killing cancer.
Yeah, the music is really quite inspirational, isn't it? So I like the idea of showing that, because in the pictures, when they're showing what's happening, the T cells are represented as just these spherical things here, but really they're very motile, and they interact with the cell that they need to destroy very intimately, so that they're not just sort of letting the cell explode like this, but rather they're getting in and sort of injecting things into the cell. And I think this picture here is pointing at the things that are released by the T cells that are going to have an effect on the target cell. And in this picture, it looks like there's this little cloud of ammunition that's sort of released towards a cell. But in the picture, the movies we just saw, it's injected directly into the cell. So it's not going to have the potential effect of harming any of the uninfected cells in the neighborhood. Granzymes are these granules that they were just talking about that have enzymes that break down components of this inside the cell. Perforins perforate the cell. They make holes in the cell, a lot like complement does for bacteria. This is going to do the same sort of thing for infected and diseased cells in your body. But those are the things that were in those red granules in the cell that moved out of the tail of the cell and towards the target cell. And then they shifted to a different sort of way of depicting things. I liked where there's that bright flash of red. Every time it kind of looked like the T cell was throwing a grenade into the cell and it was exploding inside, which was, I find that stuff kind of cool. But so that's a more realistic depiction of what the book has to depict in a drawing like this. Okay, because they've sort of picked this cartoon of a T cell. So they want this to look the same everywhere in the book so that you're like, oh yeah, that's a T cell that we've been talking about all along. But in the movie, we definitely saw that the cells are a lot more motile. Come on. Okay, so these green cells here, these are the T cells, and now the red one is in this case, the cancerous cell that they're attacking. Or going back originally, here the cancer cells in blue, and the T cells, this kind of orangey looking cell, I guess, that we saw sort of crawling along. Now, this first part of the movie, that as I was showing it to you, this is a cytotoxic T cell that they're filming. But the way that this is moving is the exact same way that macrophages and other phagocytic cells move also. And it's the same way that amoeba move. It's just the cell membrane's extending forward, and then everything follows behind if the cell decides that it wants to go in that particular direction. So this section of the book is really kind of getting at what the T cells are, how they come to be, and their types, the helper T cells, the cytotoxic T cells, and the regulatory T cells. And the regulatory T cells get a very small section of the book because we usually are more interested in the attack type cells. They're the ones that go out and do things. The next thing in the book is getting into B cells. And so this is where I grabbed that picture of the antibody I was showing you before. Before I get into types of antibodies, let me go to this picture here, actually. This is the B cell side of activation. Okay. So here are 3 B cells. Okay. They're distinguished because one has blue antibodies attached to it, one has red antibodies, and one has, I think those are purple antibodies. I'm not the greatest at color vision, so, but the color's different between the three. Those are three different cells. Now, the naive B cells, just like the naive T cells we were just looking at, they're hanging out in secondary lymphatic tissue, and they're going to be exposed to the antigen. Now, B cells don't have to use antigen-presenting cells because they're interested in the antigen in its unprocessed form. So these little triangles here represent antigens that are in the body. They've gotten into the body through a cut in the skin, or they're ingested, or whatever it might be. They're in the body, and they interact with the particular antibody that recognizes them. So in this picture, only the red antibodies bind to this antigen. There's none of them bound on the purple or the blue antibodies, okay? Because they're specific for what they recognize. That antigen binding site, antigenic determining factor, that relationship is very specific. Okay? So this particular one here recognizes an antigen. It's a naive cell, and it recognizes antigen, and it starts to proliferate, which means that clones of this cell, meaning they have the exact same DNA down to the variable region of the antibody, they start to divide. Some of them make memory cells, which, as far as the cartoon depiction is concerned, don't really look any different from that original naive cell. But there's more of them, and they can be activated to make more cells in the future. The active B cells, they start to differentiate into what's called a plasma cell. Now, in this drawing, I always kind of think of this as looking like, the end of somebody's thumb. Okay, so where the nucleus is like the thumbnail, and then the lines here are kind of like the wrinkles in the skin. Those lines there are meant to represent endoplasmic reticulum and Golgi apparatus, which are the organelles that are associated with secretion. Okay? So the endoplasmic reticulum, and its rough endoplasmic reticulum, starts to make a lot of copies of the antibody. And then the endoplasmic reticulum passes that to the Golgi apparatus, which then packages it up in vesicles to secrete out through the surface of the cell. And so the plasma cells start secreting antibodies into the plasma, which is why they're called plasma cells. So again, these are depicted as these red antibodies, just like the ones that were attached to the surface of the naive cell. But now that they're secreted, they can get all over the body and into the various bodily fluids and help to attach to antigens that might be present in our body. Okay. Now, to finish out this picture, the memory B cells that are made the first time around are available to be activated immediately when there's a second exposure to the same antigen. Okay. So back here, there's the antigen, and the naive B cell has to go through the proliferation stage to make clones of itself, and then it has to grow up a whole bunch of cells. Some of them memory cells, some of them plasma cells. The memory cells, the next time you're exposed to the same thing, are already familiar with what's supposed to happen, and they just start making a bunch of new copies of the cells. The secondary response is a lot faster than the primary response, as we see it there. Now, having shown you where the cells come from, which the book actually does second, I want to back up, as far as the book's concerned, to talk about the 5 different types of antibodies. Now, antibodies are also known as immunoglobulins. And that's abbreviated capital I, lowercase g, for immunoglobulin. The 5 types of immunoglobulins are depicted, are named, whatever, with a capital letter after the IG. So there's IgM, IgG, IgA, IgE, and IgD. Now, I want to start with IgG because if we go down here, we can see it makes up about 4/5 of the antibodies that a particular cell makes. Now, all of these antibodies have the exact same antigen binding site on the end. They're all made by the same cell for a specific antigen. What's different is kind of how they interact with each other in the at the other end of the antibody. So this is not what deals with the antigen, it's what deals with a cell, or it tags it for secretion, et cetera, et cetera. So IgG is the most common type, and they're normally found just as single antibodies secreted into bodily fluids. So really this picture we were just looking at here, all of these antibodies that are being released from the plasma cells, those are IgGs. That's what IgGs are about. They're just being released from plasma cells to do their business. Looking at the percentages here, the next most common type at 13%, which is a lot less than 80%, is the IgA. IgAs are commonly found in dimers, meaning that two are linked together. And so this little archway in the middle is just depicting how they're held together. Also, IgAs will tend to have another component wrapped around them called the secretory component, not because they're secreted and others aren't, but rather IgAs are most commonly associated with secreted fluids like mucus, tears, saliva, and then colostrum is referring to breast milk. So these are found in our bodily secretions. As far as the breast milk is concerned, IgAs are the way that a mother can transfer antibodies to the newborn after it's born. Okay. Before it's born, the fetus is getting antibodies from the mother as IgGs going through the placenta. Okay. But IgAs are passed through the breast milk. But IgAs are also found in other secretions, helping to suppress infections coming through those sites too. Okay. Then the next most common type at 6%, which is still a very small fraction overall, are IgMs. And IgMs are characterized because 5 individual antibodies all link together at the center of this cluster, which means that there are 10 antibody binding sites exposed on the outside of the IgM. And those are all going to recognize the exact same antibodies because they're all being made by the same cell. IgMs are great to help when we have some sort of cellular pathogen in our body, and 10 cells can bind up to one IgM, and then there can be another antibody bound to somewhere there, and we start to make this huge cluster of cells. Now, IgMs are actually what we use in blood typing. So when we see the clumping of cells when we're testing for blood typing, we're really using this type of antibody here. But that's an artificial presentation. We don't blood type in our body. That's just the type of antibody that's best for the blood typing reaction. The next type, based on percentage, is the IgD. There's only about 1% of these in your system. What's important is that they're associated with B cells, and they're called B-cell receptors. So if we go back to this picture here, the antibodies that are on the surface of B cells in their naive form, those are IgDs. That's what they do. Okay. They can be present elsewhere on the surface of B cells in other situations, but primarily they are there when the B cell's naive. and does its job. And then the least common type at 0.002%, so a very, very rare type, is the IgE. These will end up being receptors on the surface of mast cells and basophils. Now, mast cells are the ones that release histamine for the inflammatory response. When an IgE is on the surface of that, that means that the presence of that antigen is going to automatically cause inflammatory response. This is a fairly rare condition, a fairly rare situation, but it does happen. When it goes wrong is when that antibody is actually recognizing something that's not pathogenic. Okay? Now, antibodies recognize antigens. An antigen is just a very general term. When an antigen is something that causes disease, we usually refer to it as a pathogen. When an antigen is something that causes an allergy, we call that an allergen. And so allergens will activate mast cells directly when they bind to IgEs on the surface of mast cells. Okay. A rather rare situation. If you're allergic to things, you're probably only allergic to a handful of potential allergens. Pollen, pet dander, some molecule that's present in peanuts, whatever it might be. But it's a very rare condition for anybody to have. You're not going to be allergic to a bunch of things, just one or two things. Now, basophils are mostly associated with anti-parasitic activity, and mostly with parasitic worms. Okay, so basophils and IgEs actually might be an important part of dealing with parasitic worms, say hookworm or something like that. And since it's IgEs that are playing a part there, it's the same thing that's related to allergies, and therefore that shows us a pretty good place where the hookworm story and allergies might be tied together. Right? And again, these are very rare species of antibodies, so they don't have a real big part to play in anything. But in keeping with the Sculptors of Monumental Narrative title from the podcast we listened to, even though they're a very, very small player in the overall picture in the body, because of this relationship, it might actually be something that's very important to have how things work in our system. Basophils, I don't know too much about them specifically, but they might have something to do with helping regulatory T cells to do their job or something like that. So that might fit into the story a little bit more. I have no idea, and I'm only saying that because that links a few things together based on the podcast we listened to. Those are the types of antibodies. The rest of this chapter is going into how we have immune responses. An immune response to initial or primary exposure can be kind of minor, and then a secondary exposure is much more robust. And this picture comes right after this picture here, which is really, this is the primary response. There's more happening. It takes longer, and there's not as much of a response. But then the memory cells are there for the secondary response. And secondary response doesn't necessarily mean it's the second time. It's every other time. So the primary response is the first time you're exposed to something. Secondary response is all the rest. And they're much more robust reactions, which is described here. I described active versus passive immunity a while back, so you can see it there. And this goes into something with T cells and B cells, which you can look at. It seems like there's one more thing that I need to mention, and so I'll start off with that next time, and then we'll get into the respiratory system, and we'll move on from there. So, otherwise, have a nice day.